In vitro fertilization produces cleavage stage embryos in humans. Cleavage stage embryo then undergoes growth to produce blastocysts. The trophoblast of blastocyst is a thin layer of epithelial cells. Trophoblast helps a developing embryo to attach to the wall of the uterus. Trophoblast protects the embryo and forms a part of the placenta. A fluid-filled cavity called as blastocele is present in blastocyst. The cluster of cells inside a blastocyst is called as inner cell mass or ICM. ICM forms embryonic disc and eventual embryo proper. In the next step, the inner cell mass is separated or isolated from the surrounding extra-embryonic tissues. In the next step, the isolated inner cell mass is plated onto a layer of fibroblast feeder cells. These fibroblast feeder cells help to nourish the inner cell mass. In the next step, the cells from inner cell mass are dissociated. These are the dissociated cells from ICM. In the next step, the dissociated cells are replated. These replated cells are nourished by new feeder cells. In the next step, established ES cell cultures are formed. These established ES cell cultures can be maintained for many generations and can be stored in frozen condition. Please like, subscribe and share.